All right, hello everyone. Thank you for being here this early, as always. Good morning, Anna. Everyone in Evans, hello. Um, this site is zooborns.com. Zoos worldwide cooperate to share images of whatever was born on that particular day in their zoo. And so it's a wonderful, healthy, daily antidote to the problems that we experience. You know, traffic, midterms, those things. Not to start your Monday morning with the M word. On the midterm, I'm hopeful you will remember that humans differ genetically with respect to their ability to digest milk as adults. That the ability to do so is inherited as a dominant Mendelian trait. And I will ask you what that strange turn of phrase is. It's inherited as a dominant Mendelian trait. And I'm hopeful you will be able to tell me that that has to do with the fact that in most humans, and in fact most, if not all mammals, the ability to digest milk exists only, that's why I showed you the baby sloth, at that age. And then the gene encoding the enzyme, lactase, shuts down. But in some people, expression of that gene persists into adulthood. I'm also hopeful you will remember that the ability to drink ethanol is really ranges in frequency between groups of humans. In Northern and Eastern Europe, where I was born and raised, that ability is prevalent. And where John Belushi's forefathers was quite prevalent. And while Animal House is very funny, it really, really is, Belushi himself died at 33. He was a drug addict and quite a heavy drinker. Now, Mother Nature and humans have cooperated in this remarkable act of genetic engineering in which the ancient Chinese who domesticated rice 10,000 years ago evolved the essentially inability to drink alcohol. They evolved a form of an enzyme, alcohol dehydrogenase, that metabolizes alcohol into a toxic intermediate so fast that they can't really consume any measurable quantity of ethanol without developing severe, rapid side effects, which of course protects them from alcohol abuse. And I'm hopeful that you will also remember that for the midterm and will be able to answer a few questions for me. Now, I taught that specific set of examples, lactase persistence and inability to metabolize large amounts of alcohol. First, because I wanted to connect the notion of allele that we learned from Mendel to something that happens to you and I. Second, because I wanted to talk to you a little bit about the connection between genes and enzymes, and a good chunk of today's lecture will be precisely that. Before we dive into that, I want to tell you a story about a remarkable human being, and I actually found him by complete accident. I was not looking to teach his life history in the context of this story. And then when I looked further, I realized that he's a textbook example. He should be in textbooks with respect to one specific set of issues. That is the notion of incomplete dominance. With that, now, I unfortunately do not read or speak Chinese, so I cannot correctly read this. This is, of course, the transliteration. But needless to say, as well, those of you who know how to read and speak Chinese know that that's not at all pronounced Yang Wing. It's pronounced like that, and I cannot do that. Here is the picture of this gentleman. This picture was taken about 110 years ago. He was the first Chinese student to graduate from a U.S. university. He graduated from Yale College in 1854. And led a life of very considerable distinction, which I unfortunately haven't time to discuss because it's completely outside of the scope of this class. What is in the scope of this class is whom he married. And he married a woman named Mary Kellogg, an American. And there's Mary Kellogg's picture. And they had 2 children, Morrison Brown Young and Bartlett Golden Young. This means that their union and their children represent a marvelous exercise in the notions of dominance and recessivity. specifically, if we're making a legitimate assumption that Mary Kellogg was like her English forefathers, a certain way, and that Mr. Wing, again, I'm sorry I'm butchering his last name, was like his forefathers from mainland China, the owner of certain alleles, then this is the flag of the Qing dynasty. This, of course, is, anybody know what this is? What is that? The flag of England, thank you. That's St. George's Cross. So in that marvelous union, we had the union between an alcohol-sensitive, lactose-intolerant father, and we know this to be a high likelihood genotype for that individual because of the history of that people, with an alcohol-insensitive, lactose-tolerant mother. Now, I've written here what the Mendelian genetic relationship would be between the phenotype, ability or inability to digest lactose, ability or inability to consume large amounts of alcohol, and I've converted these into familiar to you Mendelian nomenclature, uppercase and lowercase. In the specific example of the two traits we are discussing, ability to digest milk and ability to consume alcohol, in fact, this uppercase, lowercase nomenclature is, in fact, not entirely appropriate. In fact, both from first principle considerations of biochemistry and reality, their offspring would, in fact, be alcohol sensitive. But the degree of that sensitivity, so, number of drinks one can consume before really bad things start to happen, was much milder than in that person's, and I'm really hopeful that Google didn't deceive me, and that is Chinese for father. Anybody care to confirm? It is? Yes. I was like, what if it was Chinese for something inappropriate? And then I'd be, yeah. Similarly, with respect to lactose tolerance, so ability to digest milk as an adult, the offspring of that union would be able to drink milk as an adult, but not as much as mum, which, of course, is the English form for mom. So, folks, it is time for us. By the way, I beg your pardon, I should have remembered. By way of homework to prepare yourself for the midterm, feel very free to draw out the number of gametes and different phenotypes that one would get. Let's say if two such Chinese American individuals chose to get together and have a baby. What would be the likelihood of that baby being completely lactose intolerant, intermediate lactose tolerant, fully lactose tolerant? What would be the likelihood of that baby being completely unable to drink alcohol, or intermediate, or all the way to the other extreme? But what I'd like to talk to you about right now is why some alleles are dominant, why some are recessive, and some incompletely dominant, and sorry about this, why there's also codominance. Promise, this is worth it. So, for example, an important allele of a human gene that is dominant is the allele of BRCA1 that cause mutations and which cause breast cancer, or the allele of a gene called Huntington, mutations in which cause Huntington's disease that killed Woody Guthrie. This land is your land, this land is my land. Some alleles are recessive. 1 in 2,000 live births is a baby with cystic fibrosis. They're homozygous recessive. Their parents do not have cystic fibrosis, despite both being carriers of the recessive allele. Why do they not have the condition? Why is that allele recessive? 80,000 of our compatriots have a condition known as sickle cell anemia. Both their parents have a milder form called sickle cell trait. That is a predicament known as incomplete dominance. And last but putting it mildly not least, one of many examples of codominance in human genetics is, of course, human immunological identity. Why it is unfortunately the case that folks who are mixed race have a harder time finding bone marrow or organ donors, and why I again urge you, especially if you are mixed race, to register at marrow.org because your bone marrow could save somebody's life. Really, really. Okay. I don't have time today to explain all of this to you. There's quite a bit here. So I'm just going to start with one example, which is why some alleles are recessive. So to be clear, what does recessive mean? It means that in a hybrid, let's go back to Gregor Mendel's original derivation of that word. In a hybrid, so the offspring of a union of two pure lines, dramatically differing in a trait under study. Yellow over green, round over wrinkled. In a hybrid, the trait of one of the parents recedes, hence Mendel's choice of word, recessive. The trait of the other parent is conspicuous and manifests itself in the child, is dominant. But biochemically, mechanistically, why is green recessive to yellow? Why is wrinkled recessive to round and not the other way around? Why does my microphone keep falling off? The best way I can think of explaining this to you is to remind you of that child from Pakistan whose death was tragic. However, his family illuminated a path to the development of better pain therapeutics. You will recall that this boy was completely insensitive to pain. He died just before his 14th birthday after jumping for money off a house roof. And if you look at the pedigree that that child came from, I believe it is this family. Neither of his parents had his trait. So both of them could experience pain just fine. They seem to have passed to their son an allele of a gene that in the homozygous state causes a person complete pain insensitivity. Why is that? Why is the wild type allele, as we can see in the case of his parents, dominant over the pain non-transmitting one? And the answer is a simple one, and one that I'm expecting you to please memorize for the midterm. It is that most recessive alleles are loss of function. Think about this boy's spine. In the spine is a cell called, this is not on the midterm, the dorsal root ganglion. It is the cell that transmits information about the pain sensation. If the allele of the gene does not make a functional protein, so the protein can no longer transmit the information, it will be recessive to the wild type one. Because the wild type protein will be transmitting signal, the mutant will not. So in a heterozygote, one will work, one won't. Thus, the wild type allele is dominant over the mutant one, because the recessive allele is loss of function. I see nodding. Excellent. Somebody care to tell me what this individual is suffering from? What genetic condition? Cystic fibrosis. Thank you. I'm really glad that all of you now remember the most common human genetic disorder. 70,000 people doomed to die by age 40. With the notable exception of those who have a specific mutation which can now be suppressed by this wonder drug developed by a pharmaceutical company called Vertex. The most common cystic fibrosis causing allele of this gene, CFTR, is recessive to the wild type 1. Question for the midterm. How do we know that? What is the evidence that we have from human pedigrees that says that the disease-causing allele is recessive to the wild type 1? Somebody brave enough? Alice, what do you think? Ah, smart woman. Stole for time. What is the evidence from human families that the allele of CFTR that causes cystic fibrosis is recessive to the wild type 1? Sir? It skips generation. Somebody else said something about the word parents. Young lady? That's right. So two parents can have a child homozygous for that allele. The child has the condition. By definition, because of Mendel's first law, by definition, this must mean that both parents are heterozygous for it. They transmitted the mutant allele to their child, yet they themselves are completely asymptomatic. CFTR carriers with respect to their lung physiology, with respect to their pancreatic physiology, have no disease symptoms. Why, biochemically, is that allele recessive to the wild type one? We actually know the answer to this. It's really cool. I mean, I feel weird saying cool because, I mean, there are 70,000 people who are about to die, so I don't want to sort of, you know. The biochemistry is this. The most common allele is a deletion of 3 base pairs, T, T, T. And that deletion causes the loss of 1 amino acid, phenylalanine. The resulting protein cannot fold correctly and gets stuck inside the cell. The normal protein is a cell surface protein. It functions as a channel. Question for the midterm. Why do you think the wild-type allele is dominant over the recessive one in this case? Answer for the midterm. Because it is a loss of function. The normal protein should sit at the cell surface, make a pore, and things should go through it. The mutant protein is stuck in the endoplasmic reticulum inside the cell. Thus, in a heterozygous cell, one allele makes a protein that goes to the surface and does its thing. The mutant allele makes a protein that gets stuck in the endoplasmic reticulum. What's the phenotype of the cell? Wild type or mutant in that case? Heterozygous. Wild type. Thank you, Katerina. Wild type. That's why the wild type allele is dominant over the recessive one. Sir? This gentleman, let me repeat this gentleman's very profound question, which I'm really, thank you for asking. One allele is dominant, one allele is recessive. Does this mean that the recessive allele has basically taken a vacation for that generation? Is it just sitting in a corner of a nucleus, a little room that says, all recessive alleles go there? No. It is active transcriptionally, so it makes a messenger RNA. That messenger RNA is translated to produce a protein. The protein actually attempts to do its thing, but isn't folded correctly and just sits in the endoplasmic reticulum. So the gene is active, but the gene product that it makes is non-functional. It's a loss of function. That said, the simplest way to make a recessive allele is just delete the gene. We just remove the gene. That becomes a very strange recessive allele. What is that allele called? The gene is gone. Will that entity be recessive to the other one? Of course, yes, because the gene is there. Thank you for asking. Good for you. I can now go back to this gentleman who was the first Chinese student to graduate from a US college, from Yale back in the middle of the 19th century, an impressive 160 years ago. This allows me to explain to you the concept of incomplete dominance. Now, all textbooks of genetics since about 1915 have explained incomplete dominance using flower color. I have nothing against flowers. I'm very pro-flower. So if you want to study incomplete dominance using this example, more power to you. I would like to explain incomplete dominance to you using lactase persistence. If a person has that trait, lactase persistence, then this means this person has an allele of lactase that is on into adulthood. Let me say something deep, and then you can sort of mull over it. That allele is expected to be dominant over the wild type. And here, I deliberately did not put this into the slide, so I can wake up as many of you as you can and actually give that a name. Anybody remember what was the, from a function perspective, what was the characteristic of recessive alleles? They are of function. Loss of function, thank you. Dominant alleles are gain of function. And I'd like you to please know this for the midterm. What does gain of function mean? It means that the protein produced by that allele does something that the wild-type allele does not. For lactase persistence, the dominant allele is on into adulthood. The conventional wild-type allele does not do that. It is easy to see using this example why the lactase persistence allele, the one in Usher and Taylor Swift, is dominant over the allele found in folks from mainland China or parts of Africa that don't practice dairy farming, or parts of the Middle East that don't practice dairy farming. For example, that don't drink cattle milk like they do in Saudi Arabia. It gets even more interesting, though. A person homozygous dominant for that allele, for example, the Danes, the prevalence of the lactase-persistent alleles among the Danes is like 80%. An adult homozygous dominant person would have 2 copies of the gene making the enzyme. So in that person's small intestine, there would be twice as much enzyme as in the intestine between the offspring of a union between, let's say, somebody from Denmark and somebody from mainland China. Hi, where are you from? I'm from Copenhagen. You? I'm from Beijing. Hello. And the offspring of that union would be heterozygous. I know this is silly, but I'm just trying to wake you up so you remember this for the rest of your life. And the offspring of that union would have 1 allele from Danish mom, 1 allele from Chinese dad, and would be a heterozygous. In the small intestine of that person, there would be enzyme in adulthood, digesting milk sugar, but half as much as in that person's Danish mom. This means that the phenotype, please write this down, the phenotype of the heterozygote, I love genetic words, the phenotype of the heterozygote, isn't that pretty? The phenotype of the heterozygote is intermediate between that homozygous for one allele and for the other. Sir, all the way in the back. Good for you, you smart person, you. Thank you. This gentleman is saying, good for you. But wait a minute, this gentleman says. Then wouldn't you expect intermediate phenotypes for something like cystic fibrosis? You only have half as much protein available? The very sophisticated question, good for you, implies that Mother Nature perfectly tunes output levels of its genes just for its needs. Namely, that in order for a cell to be wild type, in order for your lungs and your pancreas to be normal, you need the output of 2 alleles of the CFTR gene. That, in fact, is not the case. It seems that for some reason, Mother Nature causes even 1 allele of the CFTR gene to produce enough protein for normal function. A prime example of that is, why did Queen Victoria not have hemophilia? She was a carrier. She was a carrier of the mutant gene of the factor IX gene, mutations in which have unfortunately caused infamy for Rasputin, and thus inexplicably led him to be used as the name of a record store and telegraph. I walked into the store and said, why Rasputin? They're like, we don't know who Rasputin is. I'm like, okay, well, that answers my question. Why did Queen Victoria not have hemophilia B, even though her great-grandson, Prince Alexis, did? It's recessive. But surely that means there's half as much of the blood clotting factor in her bloodstream. Studies have shown that as little as 3% of wild-type levels of the blood clotting factor is enough for normal blood clotting. So Mother Nature, for some reason, overproduces protein off certain alleles, which is why in those cases, the wild-type allele is completely dominant over the recessive one. Good for you. Incidentally, I don't have time, it's 8:36. By way of homework, work through this incomplete dominance logic, focusing on the biochemistry. So fast allele, slow allele. Why do you think it is likely that the offspring of a union between somebody from mainland China who is highly alcohol sensitive and somebody from Eastern Europe who is one more round, why is the offspring of that union, would you expect that person to be intermediate with respect to alcohol sensitivity? Look at the slides from last lecture. Look at the nature of the allele. Why is it likely that incomplete dominance is the mode of inheritance here? In the remaining 23 minutes, I have the great fun of I have to admit to you, I love teaching this experiment. It's just great. In my mind, it's second only to Mendel in just complete. Nathalie Angier, the wonderful science writer for the New York Times, wonderfully said that the word elegant in science means something else than in conventional life. In conventional life, elegance is, hello. In science is when an experiment is not just done, but done in style. The Beadle and Tatum one gene, one enzyme experiment that I'm going to teach you now is stylish. It has a certain conclusive, elegant beauty to it. So this should be good. It's also a wonderful example of how great ideas can sit on the shelf. You know, Mendel's law sat on the shelf for 35 years. This idea sat on the shelf for 41 years. And the idea was, please don't laugh. I know this is hysterical. That genes encode enzymes. Stop the process. No, genes encode enzymes? Didn't I learn that in my Montessori preschool? Yeah, but this is 2012. We're talking 110 years ago. And back then, Dr. Archibald Garrett, who, akin to many of his countrymen, appeared to look like Dr. Watson, observed, he was a physician, but an observant physician, a translational physician. So that means not somebody who speaks two languages, but somebody who converts basic science findings into practice and findings from his practice into observations of basic science. He observed that a benign condition called alkaptonuria, which is not important at all, and yet it's in every textbook on planet Earth, because it was the first example of showing that Mendel's laws apply to humans. In 1902, Garrett observed that a consanguineous marriage is more likely to produce a child with this condition than a marriage between two unrelated individuals. Here's some pretty Latin etymology. Consangue. Those native or by learning speakers of Spanish should be able to translate that. What does it mean, consanguineous? A union between same blood, very good, individuals who are related. So, for example, a brother-sister or a first cousin union. Why would the union between first cousins or a brother-sister union more likely to yield an offspring with this rare but benign but rare condition? Garrett writes, there's no reason to suppose that mere consanguinity of parents can originate such a condition. We must seek a peculiarity, an explanation in some peculiarity of the parents, which may remain latent for generations. Ah, Dr. Garrett, you smart, smart man. Dr. Garrett wrote a book written by this rather snobby-looking fellow. The reason he looks snobby is he just invented the word genetics. The word genetics did not always exist. William Bateson invented it to describe the new science of Mendelian processes. He also invented the word allele and the words heterozygous, homozygous, and the close the door word. Oh, thank you, Mike. So he invented the words allele, homozygous, and heterozygous. If you invent that many important words, feel free to stand like this and look like him and wear a silly hat like that. Thank you very much. Bateson offers a reasonable account of a phenomenon in which the offspring of a union of two related individuals is more likely to have a particular condition. There may be other accounts possible, writes Garrett. We note that the mating of first cousins gives exactly the conditions most likely to enable a rare and usually recessive character to show itself. If the bearers of such a gamete mate with individuals not bearing it, the character will hardly ever be seen. But first cousins will frequently be the bearers of similar gametes, which may lead in such unions, which in such unions may meet each other and thus lead to the manifestation of the peculiar recessive character in the zygote. I've actually shown you an example of that, a rare human condition manifesting itself in a consanguineous union. Look at this. That was a consanguineous union, as was this. As was this. First cousins will frequently be the bearers of similar gametes. And in a consanguineous union, they are more likely to meet each other and thus lead to a manifestation of that particular character in the human being born, whether this is a child in Pakistan or a boy in London, England. Garrett published this paper in Lancet. He didn't exactly publish it in the Journal of Obscurity. This was in Lancet. And there, in black and white, it said, hello, genes encode enzymes. For example, in this specific case, there is a biochemical feature of a human being, urine turning a certain color upon exposure to air, that appears to follow Mendel's first law. Oh! And that paper, despite being reasonably widely read, just sat there for 41 years. Remarkable but true. Until this marvelous, marvelous little organism entered the equation. This, of course, folks, is budding yeast, Saccharomyces cerevisiae. Now, the experiment I'm about to describe you wasn't done on budding yeast. It was done on a different fungus. However, the core principles of genetics that operate in that fungus are also true of yeast, and that's why it's a great one to teach. A little bit of terminology, which will be useful on Friday. Why are they called budding? Does that have to do with Anheuser-Busch? It does not. Budding yeast grow by budding, so a mother cell basically buds off a daughter cell, and when the daughter cell falls off, it leaves a little scar. Oh. As I'm about to show you, yeast mate. And when they mate, they form a mating projection called a shmoo. That is, in fact, the technical term in yeast genetics. Shmooing is what yeast do when they see each other. And this has to do with a cartoon from the 50s, which you should remember if you're into obscure trivia. Now, this I need you to know, please. It's a useful and important fact and will last you a lifetime. Those of you fortunate to have read Tintin as opposed to seeing the recent movie will know that Captain Haddock has a certain problem with a certain type of beverage. Now, there's an interesting difference between the captain and Tintin and the dog, Snowy or Milou, and the organism that produced either whiskey or beer, and that has to do with how sugar is broken down. This is the core equation of humanity. We take sugar, whether from rice, which is, of course, the #1 food on earth, or any other source, and we burn it all the way to carbon dioxide and water. Yeast, Saccharomyces, the sugar fungus, cerevisiae, is Latin for beer. It gave us the Spanish word cerveza. Budding yeast ferment all sugar to form that product, which gives us beer and wine, etc., and that product, which gives us bread. The remarkable utility, as you're about to see right now, of budding yeast and other fungi as model organisms is they have a fundamental difference of lifestyle relative to us. This doesn't mean that they don't exist as diploids. They do. This doesn't mean that they don't have sex as we do. They do. It's just in yeast, the haploid phase is much longer than in humans. Humans, strictly speaking, don't really have an autonomous haploid phase. Sperm are haploid, eggs are haploid, but they're not really autonomous living creatures, which is what explains, of course, the sexual and the ploidy cycle of Homo sapiens. In striking contrast, and I should say that I wasn't able to find a good version of this figure in your textbook, so I pulled it from a different textbook. This is the one I'm expecting you to print out and please know for the exam. I'm expecting you to know about sort of the yeast life cycle, and I know that in Professor Meehan's laboratory you've done yeast work, so this should be very familiar territory to you. Yeast can grow healthily, happily, in both liquid and solid medium as haploid cells. What does haploid mean? One copy of each gene required to make an organism per nucleus in G1. They will grow mitotically with no problem. Now, they happen to exist in two sexes, which yeast biologists actually don't call sexes, they call them mating types. No comment on that one. Can you just imagine? We use that term for humans. Single male mating type seeks single female mating type for long-term relationship. Where was I? Sorry. Two haploid yeast cells of different mating type can fuse and form a diploid. That diploid can do one of two things. It can grow mitotically as a diploid. So this is the fundamental difference between yeast and humans. Yeast will grow mitotically in liquid or solid culture and form huge amounts of themselves, either as haploids or as diploids. We only grow huge amounts of ourselves as diploids, so we're all diploid. So yeast can grow mitotically as a diploid, but then that diploid cell, under specific conditions, can go through meiosis and form 4 haploid cells, each of which will go back to living as a haploid. What would I like you to know for the midterm? I'd actually like you to be able to reproduce a primitive form of this figure. Ignore the designations of the mating types. I simply need you to be able to draw the life cycle of a yeastie. Here's a haploid yeastie, growing mitotically, fusing to a different haploid yeastie, forming a diploid, that diploid growing mitotically, then going through meiosis, forming 4 little haploid yeasties which can now go their separate ways and grow mitotically. I'm expecting you to please memorize this for the midterm. The experiment that was done was actually done on Neurospora crassa, a filamentous fungus. And that had to do with a remarkable feature. Why did they choose this as opposed to Saccharomyces cerevisiae? Well, I need to tell you a little bit about their life cycle. So first, your textbook contains this rather detail-oriented version of the Neurospora crassa life cycle. It's figure 28-11. Here's the version I simplified and that I'm expecting you to know and understand. This. That it has 2 phases, haploid and diploid. That haploid cells grow, then they have 2 mating types, they fuse, they form a diploid, and then the diploid goes through meiosis, and then the result of meiosis is a sac, an ascus. Asci in filamentous fungi are one of Mother Nature's most ridiculously, insanely cool things. They have an ordered linear ascus. So the products of meiosis live in a line, in a little pod. And the order in which they appear in the sac reflects the segregation of chromosomes in meiosis 1 and then meiosis 2. So that if you take an organism heterozygous for a scorable trait, such as color of spore, you will see Mendel's first law on prime display. Those are 4 chromatids in 4 separate haploid spores from the paternal allele, and those are 4 spores with the recessive allele on the other side. So technically speaking, this is what actually happens. A diploid cell goes through the first meiotic division, and your mind should be racing now. You should be saying, aha, what separates here? The homologs separate. But what they do is they don't just fly away, bye, they go. They do that in the context of a linear tube, so they cannot migrate. And then the second meiotic division occurs, such that the four products of meiosis are lying next to each other in order in which they went through division. Now, to then confuse things, they go through a round of conventional mitosis, but this is irrelevant. This basically means that each of these spores has basically doubled itself. This means you can identify and study the product of an individual meiotic event. What would that take in a human being? What would that take in Drosophila? What would that even take in a flower? Just think about the physiology of the reproductive system. What would it take to look at the outcome of a meiotic event? Good luck with that. In Neurospora, to do that, you just isolate one ascus. Et voilà, mesdames et messieurs, je vous présente an outcome of one meiosis. This means, of course, that you can visualize in all its glory allele segregation. So that if something heterozygous for a scorable marker goes through meiosis, you can get something that looks like this, as drawn here. I need to, before explaining to you Beadle and Tatum's experiment, I need to explain to you a useful term, prototrophic. By the way, I can just sense looking at the clock, and I'm sorry, I don't mean to lie to you. I'm trying to be as honest as I can. I don't think we'll get to the quiz. But you didn't study the Drosophila cross for nothing, I promise. There will be a quiz first thing on, what day of the week is it? Wednesday, thank you. Sorry about this. I'd rather explain this to you cleanly rather than rush through this and then quiz you. But thank you for coming. I am hopeful you at least some of you found this at least slightly worth it. What does prototrophic mean? Prototrophic means not us. We are not prototrophic. Prototrophic means capable of synthesizing everything necessary from simple salts. It's easy to define something via its antonym. Its antonym is auxotrophic, needing a specific nutrient for growth. Humans are auxotrophic for all of these amino acids. They're essential for us. We cannot survive without them in the diet. In contrast, E. coli that lives in our gut is prototrophic for lysine. It can actually make that amino acid, and all of these, from scratch. Neurospora, the filamentous fungus, is really prototrophic. Its genome encodes enzymes to synthesize everything that it is made of. It will grow on simple salts like a source of inorganic carbon, potassium, sodium, phosphorus, sulfur, and one vitamin, biotin. And a little neurospora can make many more neurospores. Amazing. I present to you the Beadle and Tatum experiment. What was the question? Can one isolate mutants in biochemical processes? What's a biochemical process? Glycolysis, the Krebs cycle, gluconeogenesis, oxidative phosphorylation, fatty acid breakdown and synthesis, amino acid breakdown and synthesis, and yawn and yawn and yawn. I hear you. Trust me. Take upper division biochemistry. You will love it. What mutants have we identified so far? Around versus wrinkled. Well, which process is that? I don't know. Seed shape? I red versus I Darcy. Which process is that? I don't know. Eye color? Can one isolate a mutant in the Krebs cycle? Well, that organism would be dead, wouldn't it? Yeah. Well, what could one isolate a mutant for? They isolated a mutant in amino acid biosynthesis. How was this done? 3 simple steps. Step 1, make the mutants. Excuse me, what do you mean make the mutants? Which means zap the poor things with a mutagen. X-rays, UV rays, tobacco smoke, and the polycyclic aromatic hydrocarbons that tobacco smoke contains. Zap the poor things with a mutagen. Induce mutations. Next, and this is a cool technical step, mate these poor mutagenized things to wild-type cells, just to confirm you haven't mutagenized them into oblivion. You haven't just irradiated them to death. Obtain product of individual meioses and study them separately. Most of them will be wild type. They will not have received a mutant allele from their parent, but some of them will be mutant. That mutation was just induced. Did they have a way of inducing specifically mutations in amino acid biosynthesis? Of course not. They just irradiated the poor things, or strictly speaking, no, they did irradiate them. Next step, screen for the mutant of interest. What's the mutant of interest? Inability to grow on, quote, minimal medium. What's minimal medium? Well, it's basically medium that contains only inorganic salts and one vitamin. How do you screen for that? Well, you take your spore and you put it in minimal medium, and if it doesn't grow, good job. There's only one problem. You've just killed what you're after. Professor Beadle, I have a mutant. Where is it? Well, it's dead. What do you mean dead? Well, I put it in minimal medium, it didn't grow. Well, how are we going to work with it? It's dead now. Solution to the problem? You first grow them in complete medium. Rich food. Cheese board. Cheese board. And then you take a little aliquot and put that into minimal medium. Whichever one doesn't grow has a mutation that prevents it from growing in the absence of added nutrients. Which ones? They then screened their mutants of interest. They gave them complete medium, growing just fine. Gave them vitamins, it's still dead. Gave them amino acids, it grows now. Aha! But we know there are 20 amino acids. Does it have a defect in the synthesis of all amino acids or just one? Take that poor mutant and give it glycine, alanine, methionine, arginine. Yay! The inability, please write this down. The mutant phenotype could be rescued by the addition of one specific amino acid to the medium. Arginine. Proving that this creature had a mutation in arginine synthesis. One last slide, and then I'm going to let you go. How did they know? How did they know that the mutation affected one gene? Was it a mutation in 20 genes? Mendel's first law. They crossed the mutant spore, the mutant culture, to a wild type one. It then went through meiosis. And then they asked the progeny, what fraction of you are mutant? What fraction of you are wild type? Half were mutant, half were wild type. This simple Mendelian segregation proved that the mutation was in one gene. And they won the Nobel Prize for that. That gene encoded an enzyme in arginine biosynthesis. I'll skip through this, and I'll just show you this slide in 15 seconds. A forward genetic screen like this was done for Drosophila mutants. See if you can guess what radish and rutabaga are mutants in. Learning. They're dumb as vegetables. How about cheap date? What's that? What is the gene? It's intolerant of alcohol. How about lush? I'm not making this up. It's attracted to alcohol. Broken heart. Oh, it has a defect of heart development. How about Tin Man? Anybody care to guess? Lacks a heart. Very good. Fruitless. Male flies not interested in female flies. Ken and Barbie. My favorite mutant, and with that I'll let you go, is Ring. They had no idea what the gene did. It's a really interesting gene. I will have short office hours in 18010 right now.